Hi, and thank you for attending Open Door 2023 here at Arizona State. Today, we will be covering some of the lizard work we do, and at the end, you will get to interact with some of the models we have built. Species that are closely related and coexist often play an important role in evolution. The most famous example of these are Darwin's finches that live in the Galapagos Islands. Although these birds are closely related, their appearances can be very different, as you can see from this picture of their heads. Pay attention to the different beak curvature, length, and depth. There are many examples of this phenomena in nature, like the anolis lizards of the Caribbean or the Hawaiian silversword plants. Now that you know a few examples, can you guess what all these species have in common? The answer is that they all occur on islands. We do have a lot of examples from islands and less examples from the mainland because islands are small and it's much easier to collect information compared to large areas like the continental US or Mexico. Because it is so hard to collect information about plants and animals from large areas, we know less about how closely related species interact and their evolutionary impacts on each other. This is the question that we are studying in our labs here at Arizona State, along with Texas A&M and the National Autonomous University of Mexico. We have three specific questions that we want to answer. The first question we will study is whether species that live together with other species have different appearances, also called morphology, or whether there is no pattern between morphology and how many species live together. We will go more in depth on this question soon. Our next goal is to study what aspects of the habitat are important for each species. We do this by going to where the lizards live and measuring traits like tree height, how much grass there is, how many rocks there are, anything that a lizard might use in their everyday life. Then, we can compare across large areas to determine whether species in Mexico require the same habitat traits as species in the US. Our last question is to determine whether the habitat preferences of the lizards have changed through time. We do this by comparing the ancient climate of North America to the current climate of where the lizards live to determine where each species could have lived in the past and how this changes from past to the present. We're using Sceloporus lizards to answer our questions about the roles closely related species play in the evolution of other species. Sceloporus are native to North America. In fact, you can see them here in Phoenix, the botanical garden, and at the zoo. They're most known for the colorful bellies, which they use to communicate with each other. They also come in many different sizes and sometimes live alone with no other closely related species nearby, but other times they live with many other species. This makes them the perfect study system to answer our questions. Now, let's take our first goal and go into more detail. Remember that the goal is to study whether species that live with other species have different appearances, also called morphology, or whether there's no pattern between morphology and how many species live together. We're studying this by using CT scans. CT scans are a technology that allows us to see bones, like a three-dimensional X-ray, so we can measure the lizards in great detail. Here on the left, you can see what a CT scanner looks like for a human. On the right is a much smaller CT scanner that we use to collect our data. The steps of this are quite simple. First, we get a plastic can to hold the lizards. Next, we use a special material that allows us to tag each lizard so we know which lizard is which. Here, you can see that this lizard is marked with the D tag. Then we do this process for another few lizards and roll them up like a burrito. Keep in mind, these lizards come from museums and are not alive. Then we wrap the can in plastic wrap and then place the whole can inside the CT scanner for it to be scanned. What we get are thousands of X-ray files that are just a small slice of many lizards in the can. Then we can stack all the X-rays together to create a three-dimensional model of all the lizards inside the can. And this is what the final product looks like. As you can see, we have many lizards in one can, but we can identify each lizard by the tag that we placed on it. You should be able to see all the tags on here. After we get a full image, we can then focus on one aspect of the lizards and measure it in great detail. For example, here we have a skull where we can get much more detail and really be able to measure small structures that would have been impossible to measure in real life. Finally, we can then compare the skulls of many lizards and determine what patterns we see in nature. For example, maybe lizards that live together have to look different to coexist, or maybe there's no pattern and species can look similar when living together. Now that you know a little bit about our work, please check out the replicas of real lizard skulls by our table. Thank you for your time.